Hello. Hello, everyone, and welcome. I'm Amy Vorenberg, and I am a member of the Norfolk Country Club Speaker Series Committee. And we are going to wait just a minute while more attendees join us. We're really happy to have many members of the club able to join us, both from near here in Norfolk and far. So welcome. I want to tell you a little bit about how this Zoom webinar will work. Jessica Healy and I, as members of the Speaker Series Committee, will be co-hosting this evening. And we invite you, as the talk goes on, to use the Q&A box that you can find at the bottom of your screen. You are welcome to click on that box and type in any question that you may have. Dr. Jamie Childs has encouraged this to be an interactive talk. So if you would like a question to be answered more quickly, you are also able to raise your hand, and we'll be able to see that in the Q&A. And Jessica or I will help facilitate the asking of questions. We will get started in just a minute, but I want to take a second to say thank you to the Norfolk Country Club, to the Speaker Series Committee, to Jessica Healy as our capable chair, to Tony Kaiser, to Sandra Winberg, and to Amy Bernstein, who is our liaison to the board. And when we first met to talk about what our summer speaker series might include, we really had no idea that we would be thinking about a Zoom format. And we are doing that tonight as an experiment, and we're really lucky to have an expert among us in Dr. James Childs. Jamie is a member of the Norfolk Country Club, and we are really, really delighted to have him with us tonight. So thank you. I'm going to tell you a little bit about Jamie's background and expertise, and then I will turn this over to him. I will be sharing some slides through my computer, and encourage you to click on Speaker View so that you can see Dr. Childs as well as the slides. Dr. Childs has focused an incredibly impressive career on zoonotic diseases caused by viruses transmitted from mammals to humans. These viruses can cause serious and often fatal disease among humans and include the hantaviruses, the arenaviruses causing various types of hemorrhagic fever in Africa and in South America, the filoviruses causing Ebola in Africa, and the rabies virus, which is worldwide. He has done in situ field studies for each of these viruses, and he's also done complementary laboratory-based virological and immunological research. He has quite an impressive background for degrees, a bachelor's degrees from Washington University in St. Louis, and postdoc degree, a scientific doctorate from Johns Hopkins University in Baltimore, Maryland. And he was a fellow with the United States Army Medical Research for Infectious Diseases. He says that he's always been interested in zoonotic and vector-borne diseases caused by pathogens transmitted from wild species to humans. And we are certainly in an unprecedented moment in the world today with COVID-19. And so I welcome Jamie Childs, and I know you join me in thanking him for setting aside time today with our Norfolk Country Club community. And Dr. Childs, we're going to turn it over to you, and you'll let me know when it's time to share the screen. Welcome, Jamie.
Well, thank you, both Amy and Jessica, for putting this together. And at the last minute, when, again, my technical skills weren't want. But What I'd like to do to start off is just put infectious diseases in the last century into context. And we could have the first slide, which I'll go very briefly because it has more to do with Corona, but it's important.
Okay, hold on. Okay, and I'm going to.
Oh, you can skip that one.
Yep. Okay.
Okay. This beautiful picture is the coronavirus, and obvious why it's called the coronavirus. It has a halo of these spike-shaped proteins, glycoproteins, which stick out from the shell. And these obvious sites, which are prominent on the surface, are the actual antigenic sites by which the host's immunity fights off these viruses, both by a serologic and a cytologic response. And I just want to point out in passing, I'm a great fan of bats, but bats have been the ultimate source for any number of human pathogens such as rabies, which causes over 60,000 deaths per annum. The Henipa virus, Hendra Nipa, very important. I won't go into them, but here some you know. The filoviruses, both Ebola and Marburg, and the coronaviruses, SARS, MERS, and it's not yet proven, but will certainly be proven for COVID-19. So bats play an unusual role in the emergence of the most significant pathogens that the world has experienced. Most of these are post-1976. Rabies, of course, is one of the oldest viral diseases we can recognize in written reports going BC. And just show the next slide. So, the United States has experienced a few blips in our eventual descendancy of what infectious diseases are caused through. Now, if we look at crude infectious disease mortality, and this is from 1900 through 1996, you'll see a couple of things which happened. In 1918, 19 to 1920, there was a huge rise in mortality, well above what was the current trend. And this was due to a infectious disease caused by influenza A, which has been likened to COVID-19 in many reports. But this pandemic was on an order of another scale from what we know currently about the trajectory of COVID-19. It caused up to 700,000 deaths in the United States when the population was a fraction of what it is. And the amazing things, if we look at the United States and data from overseas, is unlike COVID-19, this virus killed healthy young adults in age groups 15 to 35, which we do not normally associate with influenza mortality. Big difference right there from COVID-19, in which the fatality ratio goes up substantially after the age of, say, 55. And I'm not going to use the word rate if I can help, because mortality rates is not the proper term, because rates have to have a temporal component to them. So just mortality ratio will do. And I've just thrown in a couple other influenza outbreaks that you can see that they caused lots of deaths, lots of fatalities worldwide. And we have no idea of the trajectory of the COVID-19 in terms of total deaths. We don't have very good data. We're not unlikely to get very good data. But we can talk about that hopefully down the line. Any questions about this slide? Oh, one other thing to note. You'll see that following the excellent curve trending towards zero in infectious disease deaths, once again, we had a tick up in 1980, and of course, everyone knows that was the beginning of the decades of HIV and AIDS. Okay, What we got next? For slides.
Okay. I went to the next slide. Can you see it?
Yeah, this is actually it's not really that important, but it shows, the top line actually shows trends in all deaths in the United States. The blue line is those from non-infectious causes, and the red line is from infectious causes. And you can see that if you consider all the deaths that are occurring in the United States, from all the reasons that people died back in that era, still the influenza outbreak shaped the whole form of the curve going well into the future. Okay, good for that one. Are there any questions yet?
Jessica, any questions?
There don't seem to be questions yet.
Okay. The natural history of coronavirus is causing severe respiratory diseases. The first outbreak of a coronavirus, which actually did cause a pandemic, traveled to over 36 countries, was that of SARS coronaviruses back in 2003. And that virus was eventually shown to be having originated from bats, in particular Chinese horseshoe bats. And during one of these wet market experiences, the viruses, which bats and civet cats and any number of species are piled onto each other, the virus from the bat actually was transmitted to civet cats. And the early genetic data from this outbreak, which occurred in Guangdong province in China, indicate the source of human infection was from a virus from the Asian civet cats, which already diverged a little bit from its original bat host. And this virus was being transmitted mostly among people during close contact. Civets are thought to have medicinal purposes in China, and people eat them and love them. And risk factors were often associated with close contact, such as in butchering animals. But once it got from animals and humans, it was being transmitted from human to human. Keep that in mind, because yes, it was being transmitted human to human, but in a manner totally different from the way that SARS was being transmitted, involving its potential for transmission among people who are pre-symptomatic. And I'm using the word pre-symptomatically on purpose. Asymptomatic patients may also transmit viruses without ever showing signs of clinical illness, but pre-symptomatic people, prior to developing their symptoms, are the ones who have the highest burden, viral load of viruses in which transmission is occurring. Then had this, 10 years later, 2012, we had this outbreak of something which was described as Middle Eastern respiratory syndrome. It was shown to be caused by another coronavirus. Now, originally, this virus probably came from Africa, from bats, but was being transmitted in Saudi Arabia and other countries through close contact with camels. So this virus had actually been present for a while. It probably has been present for many decades, because South Africa used to be a point of trade for camels being shipped to Saudi Arabia and other Middle Eastern countries. So now camels are used for a lot of things in Asian countries, racing, make great pets, I hear, but they also eat camels, and once again, butchering, close contact with blood and the organs of animals seem to play a role. Now we have the SARS coronavirus 2 pandemic, which is the virus which actually caused COVID-19. The likely suspect, I think that can be proven eventually, can rule out laboratory accidents, will be bats. Viral sequences close to those maintained in wild bats have been found in pangolins. And these animals are also butchered, and many of their products are used for food. And if you look at the panel, next, don't go back, please. If you look at the panel, second from the left, you see civet cats in the market. Then as you go over, you see camels and a possible bat reservoir. And then on the far right, you see these beautiful animals called pangolins, which have been, in many areas of Southeast Asia, eradicated. And now they're importing scales and meat from places in Africa and Malaysia into China. And we know the code viruses exist in bats in Africa. We know they do in Malaysia. It's possible they're imported by, viruses were imported along with the meat and whatever the scales. Anyway, so that looks like cast of characters, but what you have to note here, and I went on a little long, but is that in each case, an intermediate host between bats and humans plays a critical role. In the first case, it was civet cats. With SARS, with MERS, it's camels. And although we don't know if pangolins played that role, these intermediate hosts play a key role in heating up the virus and making it more likely to be introduced to human populations because of their potential role as food sources and other things, where in some cases, viruses such as Ebola may have sprung directly from bats into humans, but then human-to-human transmission takes over. And it's the human-to-human transmission what we're really concerned with, although, and why is this happening now? Why all these new viruses in such a few short?
Before we go on, I do have a few questions from the group.
Please.
The first one is, how do we feel comfortable with the data, given the difficulty in collecting information in many developing countries? You shared much about Africa and elsewhere. How do we have confidence? And maybe the answer is we're in 2020, we should have confidence, but I'm curious.
No, doubt everything. I'm not sure confidence in data means the ultimate sources, or because several of these are very confirmed, and they generally, the hypotheses are generated on the basis of molecular data in which we're sequencing the whole genome of these viruses. And so we can look at mutations, and we can compare mutations between, say, a horseshoe bat and those in a civet cat, and infer that they were the source, and with a great deal of certainty. And we have less information on the ultimate origin of the MERS virus in South Africa. But molecular biology is what we use to track mutations in viruses and infer their phylogenetic, their family relationships. And we can also use them at a very fine scale to track who's transmitting what virus to whom.
I see. Before we leave this slide, one other question.
Did I answer the first one?
I think so. That was super helpful. The second question is, why always bats? We all read a lot about how great bats are in terms of controlling mosquito and other insect populations, but why are they the carrier for this deadly disease?
Oh, boy. Written some papers on this and a lot of speculation. Bats are one of the oldest mammals to have developed as a recognizable form today. So they're very old animals. Some of these viruses which associate bats have a genetic structure which is sort of unique and found highly associated, and they're single-stranded negative sense RNA viruses. Why bats? I don't know. Rodents transmit more pathogens, zoonotic pathogens to people than bats, but with very few, they don't cause the sort of pandemics we're seeing here. I'm talking about things like Lassa fever in Africa. hantavirus pulmonale syndrome in the American Southwest. So bats are associated with many spillover events with very significant fatal viruses. But many of those, except in exceptional circumstances, such as in hospital settings, do not transfer human to human. These bat viruses are strange. They have many mechanisms which make them important. They're volant animals, so they can carry these viruses for long distances. They're highly social, so they can transmit them among their pack mates within caves or trees. The small bats echolocate, and you've heard that speaking loudly can be a risk factor for transmitting the COVID-19. And bats, if you're standing in front of them when they are echolocating at full blast, it's like standing under a jet plane taking over. That's how much force they're emitting. So if you talk about something being emitted with the force of coughing, sneezing, or in this case of bats talking loudly, they have many factors in favor of them. They also can go into torpor or even hibernate. And some viruses maintain very well in hibernating bats and then reassert themselves when bats are beginning to socialize again, providing a means for transmission over the winter, where some viruses would die out if they were dependent upon, say, a mosquito vector. Anyway.
We have a follow-on question about bats.
All right, I love bats.
All right, so here we go. Are the animals, bats, pangolins, etc., farmed or raised wild? If they are farmed, can there be ways in which their production is regulated to control the potential for virus?
As far as I know, other than zoos, no one's farming bats. Large bats, which are harvested for food, tend to follow the flowering structure of trees. They have to migrate in order to keep involved with the fruit and flowers of trees. And not all trees bloom at the same time in the same region. So bats tend to be much more migratory than, well, we know our bats here are very migratory. So they move following the food sources. And trying to farm bats, I'm sure you can do it, but may not be very cost effective because they eat a tremendous amount of food, and just feeding them might be a problem. And there's still a worldwide trade in bats. They're regarded as having delicious dark meat. I've eaten them. All in all, I'd just say I prefer pangolin. But in any case, so there still is a world market in trading these wildlife, not just bats, but all these species which end up in these wet markets where they're so cluttered together that it's a great mixing pot for viruses to expand their ranges. So that's my short answer about farming bats, and I hope I covered most of the points.
That's super helpful, in addition to the color about you actually eating a bat, which I'm still struggling with a little bit. But before we move on, there was an earlier question about an earlier slide. And so I want to bring that back just to make sure we don't miss it,
right.
which reads as follows. In the earlier graphs, what accounts for the general downtrend in respiratory death ratios after the Spanish flu epidemic, particularly after the late 1930s?
I so hope someone would notice that.
Oh, excellent.
If you look at this slide, when did our first polio vaccines become available? When did our pertussis, measles, and so forth? What you'll note here is infectious disease mortalities were dropping readily before any of these vaccines became available from the mid-50s and polio and so forth on. So we were seeing a tremendous decrease in infectious disease deaths even prior to development of antibiotics. Remember, penicillin, late 1900s, and others which took to 1940s and even later being produced in any quantity. Yet we see this incredible drop. People explain a lot of different ways, and many people just not quite coinciding, but I'll mention it. If you do a poll of physicians and you ask, what was the most important public health achievement in the 20th century and the late 19th? It was the availability of clean water. Not antibiotics, not vaccines, clean water. So along with general hygiene improvements, I can wave my hands and say, that explains it all, but people really don't know, but it's just a dramatic pattern.
That's interesting.
So that question, thank you, I guess. Anyway, why bats? We can go to the next slide. One of the things which is incredibly important is these viruses are out there. We know that. But bringing them into contact with humans can be difficult if bats are staying where bats belong. But this is an example of a pig farm in Malaysia, which was the site of one of the Nipah virus outbreaks, which I won't go into, with fascinating biology. And you can look in this karst country and see the totally virgin forest climbing up these impenetrable hills. And where people put the pig farms are juxtaposed to mango trees and so forth, which are highly attractive bats. So we've moved not only ourselves, but our domestic animal farming, don't even have to be farming, into these, what were once, not many years ago, pristine habitats. And essentially we're juxtaposing, like we do in wet markets in China, we're just providing a interesting experiment to see what vessels viruses can grow in and where they do best. So this whole phenomenon is not going to go away. We're going to see these emergences over and over again because the world's a crowded place. And we are going to continue to be right there in the center of the world, forging ahead and clearing forests in Malaysia and Amazonia, etc., and looking forward to seeing what comes next. Anyway, but yeah, this is my slide of caution to everyone out there. Once coronaviruses get in the humans, mostly transmitted from human to human, but there's potential for other mechanisms of travel. So I put the warning sign up here. If you do this at home, please gargle with bleach. Before and after, I should say. Okay. Now I was going to tackle some of the jargon, which I think everyone's been exposed to in newspaper articles. And one of the most fascinating things which we saw with SARS, and this graphic shows actually information from SARS, but it's also showing up with the coronavirus, COVID-19, is the role of super spreaders. Super spreaders are individual humans who infect an inordinately large number of persons way outside the expected number that they would generate. I'll talk a little bit about this more later. If you see here, if you follow that person number 1, he started chains of transmission. Many of them started through 2 or 3 people, but they infected other people who went on this, in fact, 3 or 4 others, but they infected this person number 6, who started a whole nother huge cluster. And this person infected a few other people. And they're infecting literally dozens of individuals. And these events have been seen over and over with coronaviruses. And whether it's the virulence of the strain, which we don't think varies much, or the conditions, whether these were in households or in hotels, which is the case here, certainly with number 1, the conditions which permit large number of people to be infected, which would include obviously indoor environments and environments with high density of individuals, be it prisons or food processing lots or cruise ships or whatever, you can see these events unfold. And with COVID virus, there's an observation that 80% of the cases are probably caused by 20% of the infected individuals. So there's no, I'll talk a little bit later about this mysterious R naught. What we can see here is these individuals seem to infect an extraordinary number, while some others generally only infect 1 or 2 or 2.5, which is the norm with most of these coronaviruses. So super spreading is a real factor and extremely important, and one of the reasons why case tracking and contact tracking is essential to try and stem these extraordinary events.
We have a question that relates to that, which has to do with spreading, which is, would you elaborate a bit more on the asymptomatic spread of the disease? If
Yes,
you aren't coughing, how are you transmitting?
Yes, I will. Let's see. Let's go to the next slide. Let's see what we have here. This just shows the relationship of various bat-like viruses, with SARS at the top, Wuhan virus in red, Middle Eastern respiratory syndrome below that. Now you can go to the next one. Go to the next one. We can always come back to this. Go to the next one, please. Okay. There's been a lot of misinformation in the press on what asymptomatic versus pre-symptomatic means. People who are asymptomatic after being infected with coronavirus, who then do not go on to develop any sort of illness, severe or mild, are thought to shed very low loads of virus and not be very important in maintaining transmission. Those are people who are asymptomatic through the course from infection to when they develop immunity. However, pre-symptomatic individuals, and remember these are really asymptomatic this time because they're not showing any symptoms. Well, pre-symptomatic means that they will go on to develop the symptoms. They shed virus at very high levels before they become ill. And this is a key point, which I will come back to explain why this is so important, why it makes controlling COVID-19 so much more problematical than, say, SARS. So pre-symptomatic individuals will become symptomatic, and they're shedding a lot of virus, and they're shedding virus days before they develop their symptoms.
And presumably that's because they're A, unaware, and B, coughing or sneezing.
Well, they're unaware because they're not sick.
Understood.
But they are coughing and sneezing and probably talking too loudly.
Mm-hmm.
And say, oh, God, I feel awful. So
I see.
those are the individuals which we are really concerned about as the major transmitting individuals. Now, is that clear? Because this is really a very important point.
Well, it's particularly interesting in that there was such confusion even by a spokesperson for the WHO this week about this very thing. So,
yeah, well, okay, let's see what I have next. Or you want to go? Are people interested in different? Okay, this just shows that very simplistically, and this is just on a gross scale, we can track where viruses originate in the United States, where they came from Italy, other places in Europe, many from China and Asia, as you see on the West Coast, but cases all over the United States, most of those in New York and places actually were seeded from individuals traveling from Europe. And same is true in Brazil and other places where relatively rich people returning home from Italy and so forth and brought the virus with them. And this is on a gross scale. I mentioned before, on a fine scale, we can track the individual virus that a person transmits to another because of the level of genetic homogeneity or diversity between those viruses. So we know track the exact traces, which means finding individuals who are sick, putting them into isolation, quarantining those who they had been in contact with is vital shoe lever-based epidemiology to control these things. Okay. Now, I just want to talk a little about different tests. And you've seen, I think, the criticism, and it's rightly justified for people who combine PCR tests with antibody tests because they tell you two different things, and they're used on widely different scales. The test we use to measure acute infections, that's when someone's shedding the virus. That's when you stick that swab up your nose or you spit in the bottle. That's a genomic test, and it detects the viral RNA in the patient sample. And that means the infection is acute, the virus is present, and can be transmitted. The serological tests people are talking about now, well, to test past infections. They say, have you ever seen this antigen before? And if you have, you've developed these antibodies to it, which may mean you're immune, hopefully they do, but it's not measuring active tests. Data have to be reported separately. One measure is ongoing active infections, other level is the measure of immunity in the population. And CDC was reporting these combined data. I have no idea how they're breaking it down more finely, but they should never be taken to mean the same thing. Antibody tests are critical and will continue to be critical to track the level of herd immunity in the population. How many people have had this virus and developed immunity? And that number, caused by natural infections, plus hopefully the number of infections which never happened because we vaccinate people, will go into determining the actual critical level of herd immunity that's required to block transmission. And if you go on, if other questions?
There are a few questions, they sort of tie back to earlier comments. One is about, again, back to the bats, I apologize. It reads, you say that bats are some of the oldest animals around. How long have these coronavirus diseases in bats been around?
No one knows, but basically looks like bats have been around in their current form, even with evidence of being able to echolocate and fly, since Gondwanaland was present, before all the continents diverged. We have no such data for viruses, and you can extract information by extracting DNA from individuals or so forth who have passed on even centuries ago. But RNA is a much tougher molecule to deal with because it degrades very quickly. So that's a very hard question to address.
Except to say a very long time. There are a couple of questions. Yeah, there are a couple of questions that relate to earlier slides. I don't want to lose them. And I will say that's a testament to the depth and breadth of the talk that you are getting all these questions, which is great. One is, are there any particular characteristics of a super spreader?
A lot of research is going into this. And I don't think anyone can nail it down to particular risk factors alone associated with the individual who's ill and shedding the virus. Other than the fact that this person is acutely ill and peers will likely be shedding very high viral loads. Now, what causes very high viral loads? There's some evidence that certainly the immunoantigens that we use that are essential, for example, predicting transplant rejection or so forth, those antigens are probably important in how people mount an immune response to this particular virus. It turns out in some studies that people who have type A blood, for example, are more likely to be prone to very severe disease and may have higher viral loads.
Right.
But then you have the context where these events associated with being indoors, were they associated with people singing, were they associated with other activities indoors which promoted closer contact? And then what was the temperature? What was the humidity? Viruses which are transmitted by droplets and even fine particle aerosols fall to the ground faster in humid weather because whatever's expressed from a cough gloms onto water molecules and doesn't travel as far as the 6-foot rule, hopefully. Right. So people don't know, but I mean, I could speculate forever, but we're trying hard and hard to get a handle on that. What actually is the unique attributes of situations where these can happen and how we can go about potentially, I say very potentially, preventing such complex situations occurring. Sorry, I can't be more.
No, that's quite all right. I mean, Amy, I think you wanted to answer some questions live. Or maybe, am I misunderstanding that? There's a couple questions that relate to testing, and I'll read them both.
Okay.
Would an asymptomatic individual test positive in a PCR test, which seems like a yes or no answer? And the other one, but you're going to probably tell me it isn't?
I'm going to say that a pre-symptomatic person will test positive. An asymptomatic person might well test positive, but these individuals sometimes fluctuate because the viral load is such that you might be able to detect it by a PCR one day or 2 days, but you might not, again, a day later, I mean, positive a day after that. A number of people going positive, negative, But with the pre-symptomatic, you definitely, or I won't say definitely because there are false positives, but you're much more likely to pick up a positive signal.
So similarly, and in the same vein, could you discuss briefly the accuracy or lack thereof of antibody tests?
Oh, Lord. It's
hand to the head sounds like
so
a
variable,
complicated answer.
But it's if you think about an antibody test, it's telling you hopefully two things. If someone's got the infection, it tests positive. If someone doesn't, it tests negative. But no tests are completely sensitive or specific. Sensitive meaning it picks up all the positive cases among those who are really positive. Specific, it picks up all the real negative cases about those who test negative. In real life, they're not perfect. So inevitably what you get are some people testing negative who actually are infected, and some people who are testing positive who aren't infected. And that's just the nature of the test. And I'll only put it into one context. If you have a test that, say, picks up 95% of the positive individuals, so picking up most of those at 100, if the prevalence of the actual infection is very low, say 3 to 5%, you can see that even if it's 95%, in this case, specific, many of those individuals out of 100, at least 5 or more, are going to be false positives. And that means that if you only have 3% in your population, the majority of people that you're coming up with positive results aren't really positive. So test specificity has to be taken in context of the actual prevalence of the virus in the population being tested. And it's a complicated answer, but as more people become infected, then you worry less about the actual confidence interval around those values. And plus, if you have a test which has stopped capturing a large percentage of the positive people, but instead giving false positive or false negative results, that could be a big problem because you're giving people a clean bill of health and they go on. So these things are really trade-offs between what might be the specificity and what might be the sensitivity of these assays to come up with clear answers about individuals who are actually infected or not. That's a complicated answer, but I hope I got to it.
Yeah, that was helpful.
I
There
hope.
are, I don't know where you are in your talk. We do have 5 outstanding questions, and I want to make sure that you have time to share what you plan to share and we answer these questions.
Okay, I will stay on as long as I want, but this is the crux of the matter. This last slide is what you need to know about epidemiology. Have many people heard about this R factor referred to in the COVID-19 news, that people are tracking the R factor live and this and that? It's actually a mathematical construct, and it's actually called R naught. You can see the symbol up there with the R and the 0. And mathematically, essentially looks at, can be estimated by looking at the contact rate, the duration of infection, and the probability of transmission per contact with an infected individual. And I'm going to normally use the inverse of duration. Say if your duration of infections is 10 days, then your infections declines by 1/10 every day. And you look at all these denominators and numerators, and they all cancel out, so you get this R naught figure. R naught is an essential parameter in epidemiology thinking in current, certainly in the last century. And what it means, what it estimates, is you take one infected person and you drop that person into a pool of homogeneously mixing, just totally susceptible individuals. On the average, how many cases will that individual cause? Okay, boil it down to simplicity. If R is less than 1, each individual's not infecting enough people to replace himself or herself. So the transmission dies down because they're not replacing each other. If it's above 1, any fraction, that means each individual's on average infecting more than one other individual's, and you get rapid growth. Now on the slide, what you see on the y-axis is the basic reproductive number. So it's estimated for COVID right now to be about 2.5. That means that each infected person with COVID is transmitting that infection onto 2.5 people. Now, you know that's not capturing everything, because I showed you the graph of the super spreaders. Some people are spreading it on to 50 people. Remember, this is an average. Many aren't spreading to anyone. And if you look at this figure, you can see that SARS may have had a slightly higher reproductive number. But look at the x-axis. It shows that about 1% of infections with SARS occurred before the symptoms became apparent. That means everyone who was infected with SARS was symptomatic, basically. So we could use true epidemiology. We could screen for fever and other things at airports and shut down the SARS pandemic, bam, quickly and conclusively. COVID-19, we talked about pre-symptomatic and post-symptomatic. We don't know how many proportion of the cases occur before a pre-symptomatic person becomes symptomatic, but it's probably way up there. And this means that on the whole, this is going to be a very, very different experience in controlling this. If you can't put thermal scanners up to people in airports, if you can't ask them if they have a headache or fever or whatever, well, thermal scanners will do that. We can't do that with COVID, but we could with SARS. And to get to the bottom line, how do we control a virus with allowing herd immunity to develop? We've gone through that term, and interestingly, in vaccine. How many people do we have to vaccinate? What proportion of the population? Remember, this is the average. And R naught gives you mechanism for calculating that. And this is the actual figures for what people say is that we need a 60% herd immunity, 60% of a population be vaccinated in order to achieve control. And it could be a combination of natural immunity as well as vaccine immunity, keeping in mind that sometimes immunity isn't going to last as the immunity required from actual infections. We can calculate that if the R naught for COVID is really around 2.5 by those little equations at the bottom, we need to have 60% of the population immunized. And what that means, when you have that, it means that in 6 in 10 occasions which an infected person goes out and contacts someone, that person is not going to be able to transmit the virus because they're immune. Only 4 in 10 encounters will result in transmission. And I'll stop there. I hope that was clear, because this is really a critical point, and you see it referred to over and over again. And I'll take the rest of the questions.
Sure, that was super helpful about the R naught factor and the 60%, although somewhat demoralizing. In fact, I can't imagine when we will hit a 60% vaccination rate in the United States, let alone worldwide. But
yes,
let
that's
that one
another
drop.
question.
There was, and you answered some of the questions in your final comment, so I'm just going to get to some that you didn't. This is an interesting question. You talked a lot about symptoms, but there's a more specific question about symptoms. It reads, how do you account for the wide variety of reported symptoms? Fever, loss of smell, taste, you're laughing, lung, respiratory, etc. And why are some cases mild and others fatal?
I will talk to some of the symptoms first. And you have to remember the symptom list has grown dramatically since the first medical clinical descriptions of COVID-19 occurred. Originally, they had cough, fever, and a few other ailments such as headache, maybe gastrointestinal complaints. But then as we saw this emerge in young adults, we saw instances of stroke increase, a very unfortunate type of stroke where some of the usual mechanisms for coagulating blood and treating strokes were not effective. And we saw this Kawasaki-like syndrome develop in children, recognized in children. So the symptoms have grown. And remember, this virus is new. And we've made great progress in understanding it clinically. Hopefully, we've made progress in how to treat some of the specific disorders, be it dialysis, because it can shut down your kidneys. Patients may require dialysis in addition to ventilated breathing. So we've learned a lot. We probably don't know all the symptoms, but I think we have a good idea. One of the things which we don't have a handle on, and this is, again, disconcerting, people who have a severe disease, it's not like you're getting up and walking around a week later. You are in the hospital for often weeks. If you're put on a ventilator, good luck. Because then there's all sorts of repercussions and requirements for therapy after coming off the ventilator. It's not something to go into lightly. So the clinical treatments are as they are. I should say that some antivirals appear to be good, remdesivir. And at one point, cats, felids are very, in some studies like ferrets and minks, outbreaks in these farms, cats are very susceptible to this particular coronavirus. And I saw that you can buy a single dose remdesivir vaccine for your drug for your cat for $10,000 online. Now, if anyone wants me to, I will forward them that link. But in any case, I think I went on. Where are we now?
We have 3 more questions, and I'm going to ask them succinctly. One is, and 2 are quite science-based, and one is more sort of your thoughts, your general thoughts. From a water expert in the community, it reads, reports on water data suggest that 1, COVID is not in drinking water sources, i.e. that our water is safe to drink, and 2, that COVID can be traced in wastewater effluent to track hotspots. Are these consistent with what you are seeing?
The answer to the second question is yes. I've read a number of very interesting reports where people who have been testing wastewater sanitation plants during the course of this outbreak, once those samples that were collected and tested for the viral RNA, they picked up viral RNA in terms of increasing loads of RNA in those sanitary samples well before clinical disease peaked. So I think there's a real role for tracking sanitary.
Well, that's fascinating.
And they're much easier to collect than getting blood samples from people.
Exactly.
And a lot of places are already instituting that.
Mm-hmm.
Now, the question of, we know that virus is transmitted, obviously, through the feces. I have no idea, I don't know anyone else has any idea how important that is to increase transmission. I think that even though these viruses may not survive on doorknobs or your hands or even your windows, if you have both the potential for contaminating surfaces through coughing up the virus, as well as contaminating surfaces if you are less sanitary than you might be, that these could lead to infections being promoted from fomites or just organic surfaces, non-organic surfaces, not humans. So, but that question is really right on. I think that many places have already implemented testing of sanitation facilities.
Right.
Thank you.
And there obviously is a history with cholera and other
Cholera, yes.
That was a water.
Luckily, cholera doesn't get transmitted through the respiratory route.
Right.
Always.
Some more forward-thinking questions, and with the population of the Norfolk Country Club, I think is a particularly interesting one. What advice do you have in terms of restrictions on activities for someone who is over 70 years old and in general good health?
Well, if you remember the Country Club, I would go out and play golf, I'd play tennis. I would not restrict your activities at all.
Mm-hmm.
However, I think that people, even though the world is opening up, ready or not, here we come. Just people have to be responsible. I think they have to take personal responsibility. I know that people over 70 do, but to protect themselves, social distancing, wearing masks and so forth, washing their hands and so forth. But remember, that's only as good as what you're doing, but you're dependent upon other people doing the same. Now, masks are important because probably it limits the respiratory burden of viruses being coughed up and sneezed up by people who are infected. Most of the masks, if they're these N95, which have very fine particulate filters, are not going to protect you from infection if the aerosol's out there. So what you want to do is at least have them reducing the amount of virus which is shed from people who have it, and with the little bit of added protection it has from people inhaling or respiratory droplets. But still, I just saw you wipe your nose, naughty, naughty. And then you wipe your eyes. Oh God, don't do this at home. Anyway.
No
So-
kissing camels nor licking your finger. No, I'm kidding.
Right. And so anyway, I think that at all age groups, level of society, hopefully people will be cognizant that their actions, while they may not be at great risk, could affect putting people at great risk and dire consequences.
Right.
Well, thank you.
I thought you wanted me to address the economic situation too.
Not tonight.
Anyway.
Thank you, Jamie, very, very much for your insights, for all of your really interesting information, and for helping us to learn more about a really important moment that we find ourselves in. We have high hopes for more programming that will come, some different kinds of programming. We'd love feedback about whether you're up for more Zoom conversations, but we're really interested in bringing together the community in innovative ways in this period. So thank you all for being here tonight. Thank you especially again to Dr. Jamie Childs. We are grateful, and to Jessica and the committee. We are really glad to have had your expertise tonight. So thanks, and have a good night, and we will see you on the golf course. Okay, take care, everyone.
Take care, everyone. Bye.
Good night.
Bye-bye.